Well, hello, everyone. My name is Don Ord. I'm the acting director of the IGB, and it's really my pleasure to welcome everyone to today's science chat on the initiative, on the Nutrition Initiative at Illinois. This is a campus-wide personal nutrition initiative that was launched just this year in July. It's a partnership between the College of ACES and the IGB. Its goal is to optimize human health and the quality of life by tailoring recommendations not just on diet history and phenotype, but also on an individual's unique genetics, microbiome, and metabolome. Sharon Donovan of the Department of Food Science and Nutrition here in ACES is the director of the Personalized Nutrition Initiative. Professor Donovan has spent her career studying early human life nutrition and using piglets as a preclinical model. Her ongoing research focuses on dietary modulation of the microbiome and host microbiome interactions affecting the brain, gut, and microbiome axis. Sharon's been broadly honored for her scholarship, including election to the National Academy of Medicine in 2017. In today's science chat, in addition to hearing from Sharon, we'll also hear from Holscher, who's the assistant professor of nutrition, and Naiman Khan, who's assistant professor of kinesiology and community health. This chat will be moderated by Elena Camphor, who is the Director of External Relations and Strategic Partnerships at the IGB. So Sharon, thank you very much for organizing the science chat, and I will turn it over to you to get started.
Okay, thank you very much, Don, and it's really a pleasure to be here to talk about our new initiative. Of course, my slides were not going forward. So before I talk specifically about the initiative at Illinois, I'd like to give a little bit of background on, what is personalized nutrition and why is it important? So again, as Don mentioned, many of us have been studying nutrition for decades, and it's been a feature on this campus almost from the beginning of the campus. But the new thought about personalized nutrition is that it goes beyond just the food we eat and how we metabolize it. It's really providing a framework that encompasses a wide array of features. So all the way from our genetics, epigenetics, metabolism, our microbiome, but also our dietary habits and eating patterns, our circadian rhythms, our health status, our socioeconomic and psychosocial characteristics, et cetera, food environments. And so we have to think about we're humans living in many distinct types of environments, and that our response to nutrition is inherently different. And that is due in part to our genetics, but also to many of these external factors, which can be broadly referred to as the exposome. So this is a nice figure from a paper by Del Toro Martin, which he calls the precision or personalized nutrition plate. And so you can see that it's incorporating many aspects. And so the thinking about personalized nutrition is that we really need to be quite transdisciplinary and quite broad in how we consider how humans select the foods that they eat, and then also how that food interacts with them, and ultimately to affect their phenotype or their health and disease. So what's particularly exciting is that NIH just published their plan for nutrition, so the 2020-2030 strategic plan for NIH nutrition research. And the idea of precision nutrition, personalized nutrition, is going to be a cornerstone of their research funding over the next decade. And this is a quote at the beginning of the report from Francis Collins. What if each of us had an individualized, actionable dietary recommendation that help us to decide what, when, why, and how to eat to optimize our health and quality of life? And again, at the outset, maybe that sounds like a pretty straightforward endeavor, but once you start digging a little deeper, it really requires that we understand, how to accurately measure food intake, individualized responses, and also the social and behavioral drivers of food intake and selection. And so immediately we think about, one of the sort of weakest links in nutrition is that much of our data on dietary intake is self-report, and we know that that can be fraught with problems. So do we need better methods to assess dietary intake? And then also in social and behavioral sciences, we've known again for decades that most people know what they should be eating, but changing behaviors, particularly once they've been established, is very difficult. So we need to give nutrition advice, but we also need to work with individuals on how to change their behavior. So this is the core concepts of the plan, and you can see that there's 4 strategic goals shown in the circles. So spurring discovery and innovation through foundational research, investigating the role of dietary patterns and behaviors for optimal health, defining the role of nutrition across the lifespan from prenatal, even things like epigenetics, generational effects through old age, and reduce the burden of disease in clinical settings. And across these goals, there's 5 cross-cutting issues: minority health and health disparities, health of women, rigor and reproducibility, data science, system science and artificial intelligence, and training the scientific workforce. So as I'll go through my presentation today, I think that Illinois is really ideally positioned to take on this challenge, and it's particularly fortuitous that we're launching our initiative at this really exciting time. This is a figure by Eric Topol, and again, showing the breadth of expertise. So the exposome, which is encompasses all of the things that we're exposed to through our genetics, epigenetics, microbiome. But then you can see imaging and biosensors and the social graph. And one of the areas that got me very excited about being at Illinois is the strengths that we have in engineering and imaging and the emerging work in biosensors. And I really think that that is going to give us an exciting edge to help us not only measure many of these omics, but more importantly, how do we integrate that data, and how ultimately can we roll it out to apps, to biosensors, not only for data collection, but that can empower people to be monitoring their own health. So again, if we can look at this pie diagram of the health factors, that we have internal factors, which are all the omics types of things that oftentimes individuals measure, but think about the complexity of integrating all of these omics. Then we have the external factors, and I've kind of put a heavy emphasis on dietary intake, supplements, bioactives, probiotics, but also what is our lifestyle? What is our physical activity in sedentary time? Where do we work? Are we living in urban or rural settings? All of these things are part of that exposome that interacts with our own unique internal factors to influence health outcomes. So this can be body weight, physical performance, cardiovascular health, cognitive health, metabolic health. So basically, these are all interacting, and we need to understand this and the interaction of understand external factors, the interaction with those unique internal factors. Again, we can also think about these factors across the lifespan, and that's one of the unique things that we're proposing for our personalized nutrition, is to have cohorts that span across the lifespan. And then also thinking about detection and integration and prediction. This is really, in my mind, one of the limiting factors that we've had in this space, that people have been very focused on maybe just SNPs or now microbiome. But it's a very narrow focus, and we also are really challenged with how do we collect this data in ways that we can do relatively non-invasively, that we can do over time. I mean, there's been huge advances in smart devices in our phones, chemical sensors. Can we be wearing patches that can be monitoring metabolites? And then how do we collect that? How do we model it? And then ultimately take that into interpretation and prediction. So data visualization, AI. And so these are where, working with what we call our colleagues kind of north of Green Street on the U of I campus in the engineering computer science realms. So now I'll switch to talking a little bit more specifically about our initiative. As Don mentioned, this is a campus-wide initiative. It was established in July. So we're very new into this initiative. It's a partnership between the Carl R. Woese Institute for Genomic Biology and the College of Agricultural, Consumer, and Environmental Sciences. But I want to make a point that this was actually envisioned as an area of our strategic plan back in 2018. So our campus had already identified that this would be an area of strategic investment prior to NIH issuing their strategic plan. And as was mentioned, I was appointed director of the initiative in July. So why do I think that we're really uniquely positioned for this? So again, we've had these historical strengths in human, animal, plant nutrition, engineering and computer science. We're really good at interdisciplinary collaboration and cognitive neuroscience and imaging, one of the world leaders. Over the last several years, we've also made strategic investments in a number of areas. The Microbial Systems Initiative focused in large part on microbiome, but more broadly in microbial systems. The clinical partnerships with the Mayo Institute, as well as the Carle Illinois College of Medicine, which is the only college of medicine that has a bioengineering focus. The development and expansion of a department of bioengineering, and then the new Discovery Partners Institute across the state of Illinois. And so new opportunities. So as I'll mention some of the healthy cohorts we're considering and having a biobank, integrating AI, data analytics, and visualization for biomarker discovery, and then also integrating our colleagues in social and behavioral sciences and in translation. How do we actually get this information out to the public and in ways that is going to be beneficial? So the vision is that the PNI, Personalized Nutrition Initiative, will really be a catalyst for transdisciplinary research and entrepreneurship, and we'll collaborate with other units to advance graduate and undergraduate education and outreach. So we're looking that these various components will work together synergistically and hopefully additively to make up the Personalized Nutrition Initiative. So one of the aspects that we're excited about, because a lot of personalized nutrition has been very disease focused, but I think that there's huge gaps in really understanding biomarkers of health. So and with advances in things like proteomics, metabolomics, microbiome, we're beginning to understand that there are biomarkers of a healthy state. And so we have ongoing cohorts in infancy and young childhood. We have a captive audience of freshmen. I said in non-COVID years, we bring 7,000 or more students to campus every year. They are all required to live in university-associated housing, and most consume their meals and dining services. So we really see them as sort of a captive audience that we can begin to enroll freshman cohorts that we can follow through their undergraduate experience, but hopefully even as alumni, we'll be able to track some of their early biomarkers with longer term health outcomes. We've been developing in food science and human nutrition relationship with the Division of Intercollegiate Athletics. So they can maybe be considered sort of a penultimate healthy population. We have many studies going on with middle-aged populations. And there's also the Osher Lifelong Learning Institute, which is a national organization, but we have a group here at Illinois, and these are older adults who are quite healthy and very vibrant. Many are former faculty members. They're still engaged in citizen science. So we think that across the lifespan, by collecting biological samples for biomarkers, we can set up a biorepository for not only current, but also future research. So we envision a translational pipeline, and so projects can come at the beginning or they can enter really at any stage. These can be investigator generated through faculty at the University of Illinois, but we're also very interested in working with the food and pharmaceutical industry that might have a specific product or supplement that they are wishing to test its impact on personalized nutrition. And so again, these are just some of the areas that can feed into this pipeline. So where are we now? You know, as I mentioned, we've only been a formal initiative since July. So we're really a lot in the data gathering, and we're spending time with meeting with campus administrators, faculty, and I'll show you a little bit of results of our survey. We're also meeting with industry partners. We've had conversations with 10 different companies, and we're putting non-disclosure agreements into place, and understanding better how they would benefit from this type of relationship and how there could be a mutually beneficial relationships built. We're talking with other universities. We spoke with TNO in the Netherlands, and of course we're talking with funding agencies. And as we distill this information, this will help us to define our structure, particularly the theme areas we want to focus in initially, and our short and long-term goals. So these are the areas that we're currently developing, our themes, generate a faculty steering committee, faculty affiliates, an external advisory committee, which will be made up of people, experts in this space, both in industry and at other academic institutions, an internal advisory committee of campus leaders. We'll also have seed grants through the generosity of campus support that will be able to identify areas of research that we can spur. And then looking into an affiliates program where companies and foundations and others can affiliate with the Personalized Nutrition Initiative and help to support the Cohorts, Biorepository, and Phenotyping Core. So just to quickly, just a couple more slides, just to show that I think that we've sort of hit on something here, and that we launched a survey. We had 72 participants, but they came from across 11 units across campus. And ACES, which houses food science and nutrition and animal science, obviously had the largest number, but we're even getting folks in engineering and then the iSchool and media and National Center for Supercomputing Applications, social work. So we're attracting faculty from across campus. And I think as the word gets out, we're even going to have greater involvement. One of the questions we had is how well are our current faculty aligning with those 4 NIH areas in the strategic plan? So we asked them, are you currently working? That's the blue. Are you interested in collaboration? So again, most of our faculty, about a third, were already working in the area of discovery and foundational research, but more than 50% of the respondents said they would like to collaborate in that area. So, the orange bar is much bigger than the blue bar. So that's telling me that there's a lot of people who are going to be seeking collaborations and opportunities. We also asked a question, we had about 70 areas, and we asked, do you have the expertise and do you want collaborations? And so I have a couple of these. The main take-home message is that actually the area of metabolism was the top area, mainly food science and human nutrition, extension, animal sciences, DNS. Others, but then as we look at who wants collaborations, you can see that the bar is, it's quite different. We're now starting to bring in folks in the iSchool, more molecular and integrative, more bioengineering. So we really think is a huge opportunity to start to sort of play matchmaker and put the faculty who have the expertise together with those who want the collaborations, and to seed that through our seed grant program. This is another area which we were, really excited about, in terms of a little bit fewer respondents, but almost an equal number who have the expertise and want collaborations. But you can see, a lot of folks in bioengineering, ECE, and computer science, also statistics and psychology have this expertise, and then we can match them up with faculty in these areas. So lots of folks in kinesiology and community health want to be collaborating with these people who have this expertise. And the last in area biostatistics, again, you can see engineering with expertise and lots of people in different departments who want those collaborations. So I'm really excited about the opportunity and the enthusiasm that we've already had, and think this, we're well poised to start to really get this initiative going. Obviously, I want to acknowledge our campus administrators who have been behind us, Susan Martinez, the Vice Chancellor for Research and Innovation, Kim Kidwell, the Dean of ACES, Jean Robinson, who is this year serving as Interim Dean of the College of LAS, but is previously Director of the Carl R. Woese IGB, and Don Ort, who did the introduction, and also the staff who, without their assistance, I wouldn't be able to get much done. So you'll hear a little bit more from Elena and Anna Keck as program coordinator, and Sonia Gridley as our administrative assistant. And so if you want more information, we do have a website. We will be adding more information to it, but go ahead and bookmark that. You can always contact us by email. And we also have started a monthly newsletter. We have, the first one is out. And so if you would like to get that, you can either email us, or if you go to our website, there's a link towards the bottom that you can sign up for that. So, so thank you very much. We'll take questions towards the end. And so what I'm going to do now, as Naiman gets his slides up, I will go ahead and just give a short introduction. So as Don mentioned, Naiman's an assistant professor in kinesiology and community health, also a member of the Division of Nutritional Sciences and the Neuroscience grad program. And he directs the Body Composition and Nutritional Neuroscience Lab, which integrates knowledge in areas of nutrition, kinesiology, and cognitive neuroscience to understand the influence of health behaviors on specific aspects of attention, memory, and achievement. And then when Naiman finishes, we'll go straight into Hannah Holscher's presentation. Hannah's an assistant professor in food science and human nutrition, also a member of nutritional sciences, and she holds affiliate programs in the Institute for Genomic Biology, National Center for Supercomputing Applications. So as you'll see in her research, she's really integrating state-of-the-art technologies and bioinformatic tools. She directs the Nutrition and Human Microbiome Lab, which integrates the areas of nutrition, GI physiology, and the microbiome. And I was particularly excited to highlight both the research of Naiman and Hannah, because they are already working heavily in the area of personalized nutrition. And I think it will give you a flavor of the types of programs that we're hoping to build in the future.
Thank you for the introduction. I also want to start by thanking Dr. Donovan for the opportunity to speak with you today, as well as her leadership for the Personalized Nutrition Institute. We all know these interdisciplinary endeavors are really massive undertakings, and we're very fortunate to have our leadership. I'm also very excited to be here to hopefully highlight some work that may give you some sense of how we may be able to contribute to this endeavor. Excuse me. So my laboratory uses an interdisciplinary approach to understand the influence of lifestyle factors on cognitive health across the lifespan. Specifically, we have focused our efforts on obesity, aerobic fitness, and their behavioral correlates of diet and physical activity to explain variance in human cognitive function. This is because in approximately 1 in 5 children and almost half of adults have elevated weight status and obesity in the United States. This is concomitant with lower physical activity engagement and poor dietary adherence as far as federal guidelines are concerned. Together, these risk factors contribute to chronic disease in the United States, and are about 90% of our medical expenses could be attributed to these non-communicable diseases. It's also alarming that poor lifestyle choices are now implicated in poor academic achievement in childhood and long-term risk for dementia in later adulthood. Therefore, gaining a better understanding of how lifestyle factors and other factors like genetics and physiological factors could contribute to both personal and public health. This work is, could be very important. My interests specifically are largely inspired by the remarkable variability that we see in humans in behavior and function, particularly as it pertains to brain function and cognitive health. On the left, you'll see a figure here of the volume of the dorsolateral prefrontal cortex, a very small part of the brain that's vital for learning and memory and higher order functions like attentional control. And what you see here is that over the lifespan, almost as soon as the dorsolateral prefrontal cortex reaches maturity, you start seeing a linear decline in its volume, beginning in the late 20s. On the right is a figure that illustrates some data we've collected internally in our lab over the last 5 years in adults. This is roughly about 300 adults. And what you see is a relationship between age and response times during an attentional control task that we know is dependent on the dorsolateral prefrontal cortex, known as a flanker task. And what we see is that as individuals get older, they tend to exhibit longer reaction times, so a slowing in response in this task. And oftentimes, when people look at these data, if you look at only the trend lines or the average of the group, what you tend to miss is that there is a considerable amount of variability among individuals as they age. If you, for example, you can see that the individuals, even in their 70s and 80s, who have volumes of DLPFC that's similar to individuals who are younger. We also see reaction times in some individuals who are older maintained similar to those individuals who are of a younger age when you look at the response times. So these data essentially are provocative because they make us ask a very important question, which is that, cognitive aging, or what we consider to be cognitive decline that accompanies aging, may not be inevitable, and particularly not for all functions of the brain. And so the question really is whether, what's the cause of this? What explains this variability in aging and cognitive function? And whether it's genetics, or is it because these individuals are perhaps engaging in behaviors that may provide them some neuroprotection as they get older? So this is precisely what we are hoping to examine with our lifespan studies, where we do some work with children, work with middle-aged adults and older adults, and even in some cases with populations with neurodegenerative disease, to better understand what factors some in an individual's control and some that are not, that may predict this particular variability. Hopefully we can leverage this for a personalized nutrition or even a physical activity intervention. So we've done several studies in this area. Many of them have been cross-sectional studies. We have done some clinical trials in children, particularly focused on examining the effects of lutein, which is a dietary carotenoid, and water consumption on cognitive function in children. For this talk, I'll focus on our work in lutein because it fits the theme with some of the data we've collected, or the secondary data we've collected in genetics. So lutein is a plant pigment or carotenoid with antioxidant properties that is found in rich quantities in green leafy vegetables and is known to preferentially accumulate across the different cortices of the brain. This slide highlights a randomized control trial that we conducted to test the effects of lutein supplementation provided via a fortified beverage in combination with DHA, an essential fatty acid, with exercise versus a placebo among 8 to 10-year-old children over a 9-month period. And this work was supported by the Center for Nutrition, Learning, and Memory and Abbott Nutrition at the NIH here at the University of Illinois. And we collaborated with experts in several areas. It gave us our first opportunity to collaborate with Drs. Lisa Renzi-Hammond and Dr. Randy Hammond at the University of Georgia. We collaborated with Dr. Charles Hillman, who was at the University of Illinois, now at Northeastern University, for cognitive control with event-related brain potentials. We also collaborated with Dr. Neil Cohen at the University of Illinois, who is an expert in hippocampal memory performance, and with Dr. Art Kramer for neuroimaging outcomes pertaining to structural MRI and functional MRI. So as you can see, this is a really comprehensive study examining the effects of a nutritional formulation in combination with exercise for cognitive function in childhood. In the interest of time, I won't be able to go over all the different results. However, this slide illustrates the treatment effects collapsed for the nutrition beverage for outcomes of macular xanthophylls, or eye pigment, on the left. This is a non-invasive measure of lutein in the eye, which is known to strongly correlate with lutein concentrations in the brain. We also measured cognitive function using a flanker task, which measures attentional control. And on the right is a hippocampal memory performance task that uses pattern separation to assess hippocampal memory. The results revealed that children in the intervention group or treatment showed increased lutein in the eye. They also exhibited lower interference in the flanker task, which meant that they were able to maintain focus when they were provided with distractions in the stimulus, and greater hippocampal memory performance. Although the brain imaging analyses are still ongoing, so these data are still under preparation, these findings are the first to demonstrate changes in both cognitive function and macular pigmentation in children following lutein consumption. However, perhaps more relevant to our discussion today, we also had the opportunity to examine the potential role that genetics may contribute to the variability in macular pigment in children. We partnered for this goal. We partnered with Dr. Microbiota-Derived in Extension, who has experience in expertise in genetics and obesity, to examine the effects of single nucleotide polymorphisms in genes that we know are responsible for key proteins in carotenoid metabolism. This work was led by our graduate students, Dr. Bridget Hannon, now at Abbott Nutrition, and Ruyu Liu, who's now at Northeastern University. These findings revealed that children who carried at least one T allele, a minor allele in a BCO SNP, a T allele in a CD36, and a C minor allele in another CD36 SNP had lower macular pigmentation. These proteins are important because they encode transport proteins that, these SNPs are important because they can encode transport proteins that influence intestinal uptake of carotenoids, and also the cleaving of the carotenoids and the process of metabolism of their usage across the body. So these findings, interestingly, were consistent with what has been observed in adults, and they're the first to also demonstrate the relevance of genetics to explain variability in macular pigment in children. So this is important going forward as we think about changes in macular pigmentation, particularly as they become an increasing target for dietary interventions aiming to improve cognitive function in children. And the idea that we may need to explore dosages a little differently and really ask the question about who we're supplementing and who we're targeting for these treatments, because it's possible that some children may not be able to demonstrate those changes in macular pigment simply based on their genetic predisposition. So partly inspired by our lutein findings in children, we recently completed a randomized control trial to examine whether changes in macular pigmentation and cognitive function are evident following consumption of a food source of lutein. So this slide illustrates the Persea Americana for Total Health clinical trial that investigated the effects of a daily consumption of Hass avocado, a fresh avocado, for a 12-week period while it's incorporated in a meal relative to a control meal. And what, and given that avocados are food, that means that they have it's really a complex cocktail and a matrix of a variety of different nutrients. They also contain a higher amount of monounsaturated fatty acids and dietary fiber. We aim to take advantage of the fact that we're working with a fruit that had these perhaps neuroprotective nutrients, and several of them, to really expand our assessment of the different systems that may be impacted by incorporation of this fruit. This was work funded by the Hass Avocado Board, and we had collaborators really in several units across campus, including Dr. Bill O'Brien for the assessment of liver fat, and Dr. Nicholas Bird for insulin sensitivity, Dr. Hannah Holscher, who you'll hear from after my talk on gut microbiome, Drs. Microbiota-Derived and our graduate student, Dr. Bridget Hannon, for the genetics and blood lipids, as well as Dr. Erdman, who has expertise in carotenoids. Through this study, we're able to really examine whether the incorporation of an avocado a day over a 12-week period could impact multiple systems of health. In the interest of time, I'll not be able to cover all the findings from the PATH clinical trial. If you are interested, however, in the comprehensive findings from the trial, you can see the references below for the cognitive findings, as well as the findings for the gut microbiome. I'll just highlight, for the purposes of today's talk, with being focused on personalized nutrition, some of the work that came out of this study related to genetics. Specifically, Dr. Hannon, who was co-advised at the time by myself and Dr. Teran Garcia, published on the aspects of avocado consumption on blood lipids, including total cholesterol as a marker of dyslipidemia. Interestingly, while we did not observe significant effects of the treatment for total blood cholesterol, and you can see that on the left slide, on the left part of the slide, there were changes based on genotype for the glucokinase regulatory protein SNP, whereby C homozygotes receiving the avocado had total cholesterol concentrations that were significantly lower than the C homozygotes in the control group. Therefore, these findings revealed that the inclusion of genetics, it allowed us to examine the effects of the randomized control trial on dyslipidemia with greater precision. And if we didn't have the genetic information, we may have concluded that there are no effects of avocados on total cholesterol, but having that allowed us to better understand who benefits from the consumption of the avocados for blood lipids. We're hoping to expand the question pertaining to the role of genetics to other outcomes of interest in the study. And as you can see, we have a variety of different outcomes. And we're hoping to expand the role of our understanding of the role of genetics across these different outcomes moving forward. And we're, of course, open for collaborations and looking for partners who may help us dig a little further in these findings. So in the final few slides, I just wanted to highlight some important collaborations that we have, I think that are particularly pertinent for translating what we're learning in the laboratory to the community setting. So, while I've shown you the 2 clinical trials we've done, this is of course limited to a laboratory environment. Our end goal is to impact real life outcomes in the community. We think that the school setting in particular could be a really, really important place where we may find a translation for our findings in the laboratory, particularly in the case where it pertains to summers. So we know in the school year, many children get up to 2 meals per day in the school setting. This can contribute to up to half of their total caloric intake. We also know that most children spend a large proportion of their waking hours in the school. So as far as physical activity is concerned, it can be, majority of that is conducted in the school environment as well. So school policies could really impact health status in children. But what happens in the summer is that oftentimes children, particularly those with limited resources, often miss out on any summer engagement programs or summer wellness programs. And it's been shown that children who miss out on summer wellness programs tend to exhibit a phenomenon called the summer slide, where we don't see them maintain their gains in achievement. We see them slip back. Sometimes we even see metabolic changes in them in terms of unwanted extra weight, fat mass, or even increase in insulin resistance during that period, in part because of the lifestyle changes that are occurring this summer relative to the fall school year. So the summer is a really important time to target participants and in terms of providing with optimal opportunities to gain in terms of cognitive health and physical health as well. So for this endeavor, we have been fortunate to receive funding from the Illinois State Board of Education, and we're conducting a summer wellness program known as iPals that will use a multi-component model encompassing physical activity, nutrition, academic achievement, lifestyle enrichment, and social emotional learning to really impact multiple aspects of health. The program is aimed towards serving children from lower income families. So 90% of the participants are expected to receive free and reduced school lunches. We are collaborating with my colleague in kinesiology, Kevin Richards, who is a pedagogist, and Alana Harris-Bird, who is at Campus Recreation, as our partners, as well as the Champaign School District. While the program this year will be administered online because of COVID concerns, we are hopeful that next summer we'll have an opportunity to do this in person. In any case, I wanted to highlight this partnership because it gives us a really unique opportunity to translate what we learn in the laboratory in really important settings, particularly in children, where we know that providing a positive influence in children at younger age may stand to improve or optimize their health as they get older. So in conclusion, the key takeaways here are that what we know is that there's a great degree of human variability across multiple health outcomes, and we know that individuals may not benefit to the same extent as others, depending on perhaps their genetic profiles, which may explain some of that heterogeneity that we see. And we need behavioral interventions that really are tailored for individuals, particularly those that may be at higher risk or may benefit from these interventions. And this is really what I believe personalized nutrition initiatives are really targeting, is to leverage that human variability to then tailor our approaches to helping individuals make the most of these, prevent chronic disease, but even optimize their health. So thank you. With that, I'll conclude my presentation, and I look forward to your questions at the end.
All right, excellent. So thank you all for tuning in today. I know there are a lot of demands on our time, and with the current situation, nothing is ever easy to kind of find the time and attention to spend time listening to a webinar. So I appreciate seeing all the names on that list, colleagues from industry, from academia here across campus, and also from government. So I'm excited to see some colleagues from the USDA and NIH that were able to join us today. So And of course, thank you to Dr. Donovan for the opportunity to speak. I want to talk a little bit more from a theoretical perspective and thinking about the microbiome and highlight some of the tools and techniques my laboratory uses in collaboration with colleagues across campus to study the microbiome and its interrelationships with health. So the title of my presentation is You Aren't Just Eating for One, and I want to talk to you about how diet and the microbiome support health. So to make sure that we're all on the same page here, again, my research program looks at diet, gut microbiome, and physiology. And the overarching goal of my program is to do research on enhancing human health through dietary modulation of the gastrointestinal microbiome. And so the microbiome is a collection of trillions of microorganisms. And I'm focused on the microorganisms within the gut, the gastrointestinal tract. And so they're primarily bacteria. However, there are also archaea and fungi present, and they're present in impressive amounts. Some interesting facts for you: there are as many bacteria as human cells, and this is just within the gastrointestinal tract. And when we back up and think more specifically about their functional capacity, so the microbiome, they have 150 times more genes than that of our human genome. And this is important because this allows the microbiome to interact with our diet in ways that we can't. So what I mean by that is when we consume certain foods, they'll contain dietary fibers. And the digestive enzymes that we have can't break those down. So instead, certain microorganisms can break down dietary fibers and produce metabolites that can support human health. And this is an area of ongoing research, and I'll highlight some of the work we've done in this area. The way we're integrating this, though, is taking a step back and also looking at not just how the microbiome is changing through dietary manipulation, but how is health changing as an outcome as well. So we do a range of health outcomes, simple point of care like blood pressure, blood cholesterol levels. We also will assess oral glucose tolerance tests, and then more sophisticated imaging techniques like DEXA, so that we can look at percent body fat within visceral adiposity and subcutaneous. And then through collaborations with colleagues in engineering, Dr. William O'Brien, we also utilize a non-invasive, FDA-approved quantitative liver ultrasound technique. And so this allows us to not only see if there's liver fat present, but how much is there? And so when we look at this circle, we're beginning to better understand, again, not just how diet impacts the microbiome, but how it's impacting health and completing this complex interaction. So again, the composition of the diet impacts digestion and absorption. So we'll have digestion and absorption occurring throughout the gastrointestinal tract. But again, highlighting in the bottom left-hand corner there, where I'm showing you the colon, what happens is fibers and resistant starch, which are found in foods like fruits, vegetables, whole grains, nuts, and legumes. Again, these are not digested by our own human enzymes, but rather they provide an energy source for the microbiome. Interestingly, though, certain nutrients that we can break down will escape digestion, and those too can be an energy source for the microbiome. And we're really just beginning to understand what that means for health. So I've just overlaid some different information here. We're showing you the microorganisms that are present within the gastrointestinal tract. And you'll see there are some microorganisms present in the stomach, although they're not very many and the density is quite small. They're going to increase as you move more distally throughout the small intestine and to the jejunum and ileum. But it's actually in the colon, so it's depicted on the left-hand side of your screen, that you're going to see the most dense and diverse microbial communities. And this is because the colon is the optimal environment for these microbial communities. It has low oxygen content, fewer bile acids, and the movement is fairly slow. So these microorganisms can break down the foods that weren't digested. So the way I like to think about this is an environment. If we look at our window right now, we aren't going to see palm trees. We're in central Illinois. Palm trees aren't going to survive well in central Illinois. But for those of you that might be calling in from Florida, You may have palm trees out your window. So again, this environment makes a big difference on what microbes can survive. And one of the main ways that we have a way that we can manipulate the environment is through diet. And so that's why nutrition and diet research is so important in the field of the microbiome. Ultimately, diet provides a source of nutrients for the gut microbiome. So let me tell you a little bit more about some of the resources and some of the work that we do in my lab. So we use clinical and computational approaches to study the microbiome. So we can do complete feeding studies. Here at Illinois, we have a metabolic kitchen. Here are some of the glamour shots from one of the avocado trial that Dr. Khan was just telling you about. But we can also provide beverages or single meals per day. And when we're collecting health data, a lot of this work is also done with our collaborators in kinesiology and community health. So we'll do simple point of care measures like blood pressure or cholesterol, and again, more sophisticated techniques using DEXA or ultrasound so that we can quantify liver fat. When we study the microbiome and metabolomics, we're fortunate here at the University of Illinois to have state-of-the-art sequencing and metabolomics facilities. Indeed, the data quality we get, the access to the newest technologies that we have is just outstanding. And this is through the Carver Biotechnology Center. And so we can do 16S sequencing to see what microbes are there. But again, we can also do microbiome sequencing. So we're sequencing the entire genome of these microorganisms that are present in the sample. So in my case, we're studying the gut. And so this allows us to see what microbes are there and what genes do they have, which will allow us to see what functions that they have. So can they break down certain dietary fibers? And then metabolomics, we can do both targeted, like short chain fatty acids, or untargeted metabolomics. And these create massive data sets on the order of terabytes. And so one of the other things that we're able to do is utilize the computational and statistical resources here on campus. So we'll partner with HPC Bio, which is part of the Carver Biotechnology Center, the National Center for Supercomputing Applications. And I also work closely with colleagues in statistics and computer science to be able to convert these terabytes of data, these A, T, C's, and G's, into usable information. And so there's a lot of exciting work going on in that area. And again, the resources that we have available here at Illinois make this work possible and very exciting. So a little bit more flavor about some of the things we're interested in from the microbiome and how this might work its way out from a personalized nutrition perspective. So some of the metabolites my group is interested in are short chain fatty acids, which are derived from fiber fermentation, and secondary bile acids, which are derived from microbial metabolism of primary bile acids, which we actually secrete to help emulsify our fat. And we're interested in these metabolites because they have impacts on inflammation and absorption of the dietary components and the pH of the gut. So if we take inflammation in a bit more information, it's really interesting because the actions of short chain fatty acids and secondary bile acids can influence inflammation. So on one side of the coin, short chain fatty acids can actually reduce systemic inflammation and help reduce the risk of insulin resistance. However, there's emerging evidence, and we're delving further into this area, that secondary bile acids are increasing inflammation and actually increasing the risk of insulin resistance. So overall, my group is interested in health outcome measures related to adiposity, gut health, and insulin resistance. And I'm also going to talk to you briefly about some of the work that we've done to move this out to be able to understand biomarkers, so using bacteria as biomarkers to predict diet intake, and highlight some of the work that I'm doing with colleagues at the National Center for Supercomputing Applications to move us into more of a personalized approach for understanding these host microbe interactions. So I'm not going to go into any of the details on these studies, but I do want to say that diet affects the gut microbiota. And so we've published on consumption in humans, a randomized control trial on avocados, on broccoli, on oats and barley, almonds, and walnuts. And we've shown that each of these foods, because they contain dietary fiber and also unsaturated fatty acids, can influence the microbiota. But what we've been interested in is taking this work a little bit further and seeing if, can we predict which of these foods someone ate using the bacteria? So, and the other way we've been doing it is following up on the metabolic health. So we've been following up on both the nuts, so the almonds and the walnut trials, to begin to better understand how nuts, the microbiota, and health are interrelated. So thinking about the gut as an environment and the bacteria and the secondary bile acids and the short chain fatty acids, all of these have local effects. And some of this work is facilitated through collaborations in animal sciences. So Dr. Jason Ridlon is an expert in gut microbiology, specific bile acid metabolism. And so he and I have been working together to move this research out of the gut and to think about it systemically. And so one of the ways we're interested in this is thinking about liver health. And so furthering our transdisciplinary collaborations with Dr. Bill O'Brien in engineering, we can use a non-invasive liver ultrasound technique to assess fatty liver disease. And so you can see these different metabolites are both positive and negative regulators of liver health. And if we want to move this out even further to again be able to better understand how the diet and the microbiome influence systemic health, we can then involve colleagues in kinesiology and community health. So Dr. Naiman Khan and Nick Bird, so we can understand these relationships with inflammation and insulin sensitivity. So this work is ongoing. Hopefully we'll kick back up more in the spring once things get a little better from the COVID situation, but supported through the USDA and also the Almond Board of California. So the other work that I alluded to that I'll just briefly be sharing with you is about bacteria as biomarkers for predicting food intake. And this, again, is a transdisciplinary approach with my colleague Roisin Zhu in statistics and our work with Colleen Bushell in the NCSA. And so what we've been doing is using advanced computational approaches to do feature selection to select a compact set of microorganisms that can be used as biomarkers, doing additional classification techniques like random forest and feature selection so that we can, again, determine which foods an individual was eating. And so what we were ultimately able to demonstrate is using just 15 taxa that you can have between 70 and 96% accuracy on identifying which food these individuals consumed. And so our overall accuracy was up to 85% for this model. And this is encouraging because, as Dr. Donovan mentioned in her introduction, one of the main gaps and one of the main needs we need in nutrition research are additional objective measures of dietary intake to complement our current techniques. So these are some promising approaches. And so we're also following up on this and quantifying and visualizing the dietary effects in personalized nutrition. Roisin and I are National Center for Supercomputing faculty fellows, and we're working together to, again, take this high dimensional data, thousands of rows of information, terabytes of microbiome data, metabolite data, and then using feature selection approaches to narrow these down into more compact feature sets, so that we can, again, rank these. What are the most relevant features? What could be used as biomarkers so that we can, again, have this integrative model to ultimately have some biomarker selection for the use of personalized nutrition approaches. So lastly, some visualization. So again, working with the National Center for Supercomputing Applications, here's just a mock-up of some of the ways that we can move towards visualizing microbiome data. Because again, it starts as terabytes of ATCs and Gs, but clinicians and dietitians need usable ways to integrate this data into their practice so that we can use it in personalized nutrition. And so you can see this figure is showing some abundance and diversity overviews. And then this figure is an OTU tree explorer. So we can visualize how the microorganisms are related to each other. So I hope in my presentation, I've been able to give you a flavor and an understanding of how diet impacts the gut microbiome and health, and see how there's ongoing research going on here at the University of Illinois on the microbial underpinnings of these health effects, and that there's promising work that bacteria can be used as biomarkers and they show promise in precision nutrition. And so we're very excited for this initiative and engaging, and look forward to additional collaborations with individuals tuned in today. So thank you for your time and attention.
Thank you, Hannah and Naiman and Sharon. If you have questions, please type them into the chat area, and we can ask them. We did have one question come in already for Professor Donovan back in the beginning. I think this set of talks really helped to show the range of research topics, or just even a snippet of the research topics you are developing. But the question came in, Professor Donovan, if you could describe a little bit more about the different themes that you'll be including in the Personalized Nutrition Initiative at the University of Illinois.
Yeah, that's a great question. So again, building on some of the strengths that we have in, you know, the area of cognition, which was begun with these, the Center for Nutrition, Learning, and Memory, funded by Abbott, that was really brought nutrition and cognition and behavior together. So we'd like to build upon that. Obviously, some of the work that Hannah just mentioned in the microbiome space, because we need to understand our kind of organ of our microbiome and how that's impacting health. But I think a lot of it is going to be this integration of data. But part of why we're gathering the themes, I have some ideas, but I really want to see not only where the faculty are going to lead me, but who wants to fund certain areas and how can we engage with outside groups. But those are two that I'm pretty certain will be part of it. But we'll certainly try to also align with the NIH strategic areas as well.
Sharon, that's a good answer because the question came from outside the university. If anybody else has any questions, let us know, but please remember you can always reach the Personalized Nutrition Initiative at personalizednutrition@illinois.edu, and you'll be hearing more about it. And I know that the researchers will be reaching out to you. So
yes.
Any other questions or comments?
Well, just signing up for our newsletter. So like I said, we've issued the first one, but we're planning to do those at least monthly. And also this is a great science chat through the IGB, but one of the thoughts that we have is through the Personalized Nutrition Initiative to plan to continue to have these webinars, and they may be, you know, we may focus on specific areas, particularly as the themes develop. So, you know, sign up to be on our email list, and so maybe we'll do one all on cognition or one on, you know, data visualization, AI. We have some really exciting work going on with biosensors and through our bioengineering, and a lot related to cancer, but I think that we have real opportunities in personalized nutrition as well.
Thanks, Sharon, and thanks to each of you for participating in this science chat at the IGB. We have another question come in if you have a moment. How might this synergize with the Microbial Systems Initiative? And can resources be shared between the initiatives? That's from Jeff Woods.
Thanks, Jeff. Well, definitely, as I mentioned, I think that the microbiome is an important component of this. And so in working with Carrie Vanderpool, you know, thinking about how we might synergize. I mean, there's obviously research on campus. One of the things we just did, for those outside the university, we just did a major hiring initiative through the Microbial Systems Initiative, bringing young faculty to campus in the microbiome space. And many of them are interested in diet and analytics, and so we'd like to incorporate them. But you know, part of the initiative, so the funding that I've received from campus will be funding these seed grants, and we're hoping that, you know, what we want to do is put people together who haven't worked before, and, you know, have them be doing novel research within the realm of that. So, you know, Carrie and I will share resources where we can, but I'm sure that some of the seed grant money through the P&I will be funding microbiome research as well.
One more question Professor Donovan, in your opinion, how do you imagine patient care will change when personalized nutrition becomes a standard for hospitals and primary care doctors? What gaps do we have to bridge to make that a reality? And that's from IGB fellow Cecilia Gentle.
Yeah, that's a big question. You know, I think that's you know the fourth aim within the NIH strategic plan, and there's a lot of work going on in the area of electronic medical records. I think, you know, part of it, we have to get a handle on this variability, and we need to be able to have point of care diagnosing. I was just on a call this morning with Brian Cunningham on how do we take some of the things that we're doing in the laboratory and take them to the bedside, in my case for preterm infants. But I think that that's part of it. But, you know, Illinois sort of has got a head start on some of these areas already, you know, through our Mayo Illinois Alliance and others, where we've been working in big data and doing exactly what you're saying. So we're not here to recreate the wheel. We're here to leverage all the incredible resources that we have. You've heard about the NCSA, the Beckman Institute, the IGB, these alliances. So we'll be looking for every opportunity to take what's been built and expand it into the personalized nutrition space.
Thank you. It says 1 o'clock on my clock. Thanks to everyone again. And the session will be available online too through IGB's website after the recording is finished, so you can share with your colleagues. Okay. Thank you all, and have a great afternoon, and we look forward to hearing more about the initiative.
Thank you. Bye-bye.